And bonjour. This is Bruno Larvol. I'm here with Dr. Judith Pérez. And this is the second episode of Larvol CLIN, where we essentially are building the CLIN product in public with all of you. So thank you for joining. And please ask questions and make comments. We always appreciate it. Larvol is an oncology data intelligence company. We sell cancer data to mostly pharmaceutical companies, and we make that data also available for free for non-commercial use. And our leading product is called CLIN. CLIN is a Wikipedia of published cancer data from clinical trial. Judith helped build CLIN and focused in particular on one feature of CLIN, which we call the landscape, meaning comparing relevant clinical trial, usually in a specific population. And I'm going to ask her to walk us through that feature, and we're going to talk about it. So as you listen to us, ask us questions, interrupt us, say hi. We always appreciate it very much. Right. So, Judith, welcome to the podcast.
Thank you very much, Bruno. Happy to be here. And then let me start sharing screen to get a look to this. So what are we exactly talking about? That it's CLIN landscapes. Please confirm. Can you see my screen fine?
Sure.
Okay,
Not
thank you.
yet. No, I don't see it yet.
Okay. See.
Yes, here we are.
Now you can see. Okay,
yes.
Thank you. Okay, so yes, as Bruno was saying, we are here in CLIN where we have trial-centric data. So we have these trials, conferences, products, all the Kaplan-Meier plots around those trials, forest plots, outcome data, influencers, staff, and the product that we're going to focus on today that are landscapes. So let me go there. And in here, the idea behind this product was to compile all the data that we have structured in CLIN for a specific KPD. That's a key patient definition. So around second-line positive, HER2 positive, second-line plus HER2 positive metastatic breast cancer in here, focused on ADC environment. Then also we have other listed landscapes in here that we can check out. And the idea, let's go first to one of those, would be to have all this data that we have in CLIN in, first of all, data summary structured, where we have all the trials that are in this KPD, and we compare them at different levels. We can see the description of the trial in here based on with the intervention, control, status, and its primary outcome. In addition, in here, we have chosen the three ADCs that are of most interest in this KPD. And
just
we
to
even
interrupt,
have,
because
sure,
our audience is probably not familiar with our acronym. So we use the term KPD. Really, the idea is a key population. It's a key population definition. So a key population is not just a tumor type. It's a tumor type further qualified by usually a stage, a line of therapy, sometimes also a prior treatment, biomarker or molecular information, and sometimes additional modifiers as well. And sometimes that population is also further qualified by only looking at a certain modality or MOA, in this case, ADC. So the landscape is about, I think it's too small on my screen here, second line, HER2 positive, metastatic breast cancer, and focuses exclusively on leading ADCs in that key population.
Correct. Thank you very much, Bruno. Okay. So yes, to keep on that, like with these ADCs, indeed in particular, we have a small summary of their target, payload, and the type of linker they are featured with. And then we have in here the list of trials that will be using or evaluating those ADCs. So for the case of Kadcyla, we may have Teresa and Emilia, and so on. Different Destiny Breast for Enhertu, and Jivatko we have Tulip. And right below, what we have is a summary and a compiled table with all the results around OS hazard ratio, so different outcome values, median OS in months values, PFS, hazard ratio, median PFS, as well as some response rates, ORR, CR, and PR results. And all this data, first of all, has been manually curated, and the trials have been manually also picked based on what is the most leading research going on, and although also even marketed or compounds that could be of interest and state of the art. Then all this data, especially for those outcome data on median values, you can check it out also under the KM plot tab, where also we have in CLIN, we have digitized all the KM curves that have been published either in papers or in different conferences. And we have digitized all this data and make it available in this KM plot that is compounded of all these median values around these trials.
So,
yes.
Judith, these Kaplan-Meier curves, are they for the main population, the intention to treat population, or are some for subgroups? And do you have flexibility in looking at, let's say, subgroups for these trials?
Yes, indeed, for right now, these pre-selected Kaplan-Meier curves are for the ITT, for the main population to treat. And they have been, again, manually selected based on the latest also piece of data that is available. And nonetheless, you even though you have this pre-selected Kaplan-Meier curve, you are able to play around in here and select the other population available or pieces of data that have been digitized in CLIN. So you can modify the plot according to your needs. And even in here, you can display and see what are we calling out the different curves. So in this case, the one that is solid, it goes for ADCs. So general solid would be experimental arm, and the hyphenated one, or the dotted one, sorry, it would be the standard of care or the control arm. And you can hover over and see the different median values and the confidence interval or the n size. And right below, you have the list of trials that has been selected. Okay, if we move on to the and you have in here for OS values and PFS.
I've got another question. If you click on the eye on the right, do you get to the specific KM plot for that trial? Okay.
Exactly. You would get to the original data that has been digitized by CLIN.
I understand. And can you go there to the then original image from there?
We can track in here we do track the source, and you could get in here and go directly to the original publication and check out the original KM plot that was published there.
Okay. And then you also have the hazard ratio under
yes,
this,
exactly.
Right?
I was gonna get there, that you have the curve details where you have the two arms and the number of patients, as well as median, and at the bottom also the hazard ratio for this particular trial.
I'm particularly proud of the work that you and our engineer in France, Jean-Michel Bernard, have done in automatically extracting the at-risk table. And that's under the table here, under the KM plot. And it's done in an automatic or semi-automatic fashion. I think in this case, it's automatic. That's what it says under the table.
Instructed.
I think what
Good catch.
we will do soon is that we will more precisely align the table and the plot, which is typical, right? Right now, it's not aligned. The months of the plot do not align with the interval, which are actually months as well, of the table.
Yes, that would be a good move to really properly see which are the different time points with the at-risk data. And then, if I may continue, the next tab would be forest plot. And in here, the same data that we were seeing in the summary table, we have it in the forest plot format with the different trials listed in here. Again, you can switch from the different endpoints, OS, PFS, or others if of interest. And you see which values have been, which trials have reported significant OS hazard ratios. And again, as previously, we can play around with other data that we could be interested in, even different subgroups that we have also data available in here in a structured way. Okay.
I've got a question as well. So it's like a mini meta-analysis on the fly, right? So we provide the overall diamond at the bottom, and we can see that across these trials, there's a combined hazard ratio or a overall hazard ratio of 0.73. Yes.
Yes, correct. You can even download this data or share it in social media platforms or via email to your customers or partners. And then moving on, we have this milestones tab, where in here we have in the timeline, placed in the timeline, the different trials that we were looking at with the milestones based on clinical outcomes data or data releases or regulatory processes. And even if the trial was registrational, we have this big outline. So if we click on this dropdown, we can see for the Emilia trial, which started on 2009 and finished on 2015, we see the different milestones that were compiled by CLIN. And let's get to this later trial. For example, this Destiny Breast 03. In here, we also have captured phase 3 data releases at different time points. And we have this timeline structured here around these data releases. And we get the trial from trial initiation to completion and all the data releases that may have been going on. Okay. And if it's okay, we can move to the Twitter reactions page, where in here we trace back over 300 KOLs with their reactions or comments around these different trials. And we even have a sentiment analysis around those reactions or comments around this, whether they are, and this is LLM driven, and we may have negative rated reactions, not rated, positive ones, and even those that can be practice impacting. Yes?
Let's see the negative comments on tulips on the right, for example.
Yes.
And then we need to wrap up.
Okay, sure. Okay, so yes, not really good comments around it, probably because the PFS, the OS did not met its endpoint, or it wasn't significant really. And yes, PFS, it was, though PFS was its primary outcome, the OS benefit was not shown. So yes, indeed, this one is in Spanish. Yes, no OS impact. Yes, like all around even PFS was met, the OS did not meet its expectations. So probably that's why it's been on hold.
Understand.
And FDA has not approved it.
Okay. Okay. Well, we out of time, but Judith, you build with your colleague a terrific feature with landscape, and we have already some customers using it. But if you see this and if you're interested, if you'd like a full demo with more time to show the various capabilities that we have, please connect with us. Leave us a message on social media or just reach out directly to us. Thank you very much, Judith.
Thank you very much, Bruno. Have a good day.